Hello, welcome back to my channel, Bio with Shansi. In today's lesson, I want us to go through diffusion. Remember, we are still covering the AS Cambridge syllabus, and diffusion is under topic number 4, cell membrane and transport. In today's lesson, I want us to look at diffusion. What does it mean? Factors affecting diffusion, the difference between simple diffusion and facilitated diffusion. So let's get started. When you look at a cell, we have already seen that a cell is surrounded by a cell surface membrane. And we have seen that the membrane is partially permeable. Now, a cell usually needs a constant supply of substances, and sometimes we need substances to move out of the cell, like wastes. So the question is, how do those substances enter into the cell, and how do they leave the cell? How do they cross the cell surface membrane? And one of the ways in which molecules or substances move in and out of a cell is diffusion. So let's look at this cell. Assuming we are looking at a molecule such as oxygen, you could find that outside this cell, there are very many oxygen molecules. Those are 1, 2, 3, 4, 5, 6, 7. And then maybe even inside, there are also oxygen molecules. Only 2. So the outside of the cell has more oxygen molecules. The inside of the cell has less oxygen molecules. So that takes us to the definition of diffusion. With diffusion, we are looking at molecules being able to move from their region of high concentration. Where are we having the high concentration? It is outside the cell. And then we also have a region of low concentration. So inside the cell, the concentration of oxygen is low. Outside the cell, the concentration of oxygen is high. So you're going to have a net movement of oxygen molecules into the cell down a concentration gradient. So when someone talks about the word concentration gradient, a gradient is simply a difference. An example, outside the cell, we have 7 molecules of oxygen. Inside the cell, we have 2. So the difference between the two regions, as far as oxygen concentration is concerned, is by 5 oxygen molecules. So that means that is a big difference. So we are always going to tend to have molecules moving more from their region of high concentration to their region of low concentration. It doesn't mean they cannot move from low to high. This will still happen, but this will be less, and more of the molecules will be moving from their region of high concentration to their region of low concentration. That is why we use the word net movement. So if you look at the definition of when you look at the definition of diffusion, we can come up with a definition. It is the net movement of a substance or of molecules or of ions from a region of its higher concentration to a region of its lower concentration. So why are these molecules able to move? They have kinetic energy. They just move about randomly. So because of the kinetic energy that they have, that is why they are able to move. And don't forget, this process of diffusion is passive. It's a passive process. What does that mean? It does not require any energy because the molecules already have their own kinetic energy. So they just move about without the need for energy from the process of respiration. So in diffusion, molecules always move down a concentration gradient, which simply means they move from their region of low concentration to their region of, they move from their region of high concentration to their region of low concentration. And these molecules always tend to move up to when we have an equilibrium reached. What does that mean? It means that they move up to when both regions have the same number of molecules. So that is what diffusion is about. So within a cell, we are going to have different molecules crossing its cell surface membrane. Now, some molecules are able to just pass through the cell surface membrane. They can pass through this hydrophobic core of the cell surface membrane by diffusion, like the oxygen. Oxygen is non-charged. Even carbon dioxide is not charged. So for them, they can cross the cell surface membrane by simple diffusion. Water is polar. However, water molecules are very, very small. So they are also able to cross the cell surface membrane without being repelled by the hydrophobic tails of the phospholipids. However, other molecules that are charged, other molecules that are polar might not be able to just cross the cell surface membrane by simple diffusion. So when you look at the hydrophobic molecules also, I had forgotten the hydrophobic molecules, such as cholesterol, they can also be able to just diffuse through the cell surface membrane. Why? Because they will also not be repelled by the hydrophobic tails. So we have factors that affect diffusion. And the first factor is what we call steepness of the concentration gradient. Here we are looking at the difference in concentration of the substances. Assuming I have region A and then I also have region B, let me try to explain that concept. When you look at region A has these molecules, which I'm representing using these circles. So we have these molecules, and they are 1, 2, 3, 4, 5, 6, 7, 8, 9. There are 9 molecules, whereas region B has 2. When you look at the difference in concentration in both regions, we are going to have the differences by 7. I could have another region still, maybe C and D. For region C, it has these molecules. 4, 5, 6, 7, 8, 9. It also has 9 molecules. But then region D has 1, 2, 3, 4, 5, 6, 7, 8. So the difference here is just by 1. Now we can say that this side, these two regions have a low concentration gradient. The difference in concentration is low. So they have a low concentration gradient. But the other two, we can see that the difference is high, which we can call steep concentration gradient. So if diffusion is to occur in both sides, you're going to find that diffusion is going to be faster in this section compared to this other section. Diffusion is going to be faster here because we are going to have a steep concentration gradient. So the steeper the concentration gradient, the steeper the difference in concentration, the faster the rate of diffusion. So you're going to have more molecules moving from A to B than from B to A. And then here we shall still have diffusion, but it will be slower because the difference is very small. The concentration gradient is very low. So that is concentration gradient affecting the rate of diffusion. So always molecules will be diffusing faster if there is a steep concentration gradient, and they will be diffusing slower if there is a small concentration gradient. So the steeper the concentration gradient of substances across a membrane, the faster the rate of diffusion. The next, this diagram still shows us a concentration gradient. You can see this side has much more molecules than this other side. So obviously the rate of diffusion of these blue molecules is going to be very fast because of the difference in concentration, because of the concentration gradient. The other factor is temperature. Yes, temperature affects diffusion. If you want to check whether this concept is right, get yourself 2 tea bags. Place 1 tea bag in hot water at home or at school, wherever you are, then place the other tea bag in cold water. For the two bags, find out where we are going to have the tea spreading faster. Where will it spread faster? Will it be in the cold water or in the hot water? So when it comes to temperature, temperature is another factor that affects diffusion. The higher the temperature, the more the molecules are able to diffuse. Why? Because they gain much more kinetic energy. So they move about faster, and that is how the rate of diffusion also increases. We have another factor, which is the nature of the molecules. Now, some molecules are large. Large molecules tend to diffuse slowly, and small molecules tend to diffuse faster. So large molecules will require more energy to move. So they tend to always have a slow rate of diffusion. Small molecules always tend to have a higher rate of diffusion. We can also look at the nonpolar molecules, such as glycerol, such as steroid hormones. Such molecules usually diffuse faster. Why? Because they can easily be able to pass through the cell surface membrane because they are not going to be repelled by the hydrophobic phospholipid tails. So if they are crossing the cell surface membrane, they will be able to cross faster compared to the polar molecules that cannot cross the cell surface membrane. One other factor affecting diffusion is the surface area across which the process of diffusion is taking place. You've learned about microvilli as you were learning about the cell structure. So some cells have microvilli, and these microvilli increase the surface area for diffusion. So if molecules are this side and they are to enter into the cell by diffusion, they are going to have a lot of space where they can pass. That is what we call surface area. They can cross through here, cross through here. So we have more room where they can pass and enter into the cell. But we can phrase that by saying that we are having a large surface area over which the molecules can diffuse, compared to a cell which does not have microvilli. A cell that doesn't have microvilli, yes, we'll have where molecules can pass, but the surface area will be less compared to this cell whose cell surface membrane is folded to form microvilli. So the surface area across which diffusion is taking place can also affect the rate of diffusion. Can also affect the rate of diffusion. So those are the major factors affecting the rate of diffusion. And that is what simple diffusion is. Simple diffusion does not require energy. Now, let's look at another type of diffusion, which is facilitated diffusion. Facilitated means that sometimes the molecules are going to need help so that they can cross the cell surface membrane. So which kind of help are these molecules given? These molecules are going to need special proteins that are going to provide regions where they can pass so that they enter into the cell. So they need transport proteins. Like here, they're showing you the diagram of a cell surface membrane. And we are having a transport protein. Now, most of the time, these transport proteins allow polar molecules to pass through. Since polar molecules cannot pass through this hydrophobic core, we are going to have special regions which have hydrophilic channels, hydrophilic areas where polar molecules can be able to pass and enter into the cell. So such a type of diffusion is what we call facilitated diffusion. And why are these molecules passing through such proteins? It's because, number one, they are large and they are polar, so they cannot diffuse through the phospholipid bilayer. Even ions are also charged. So examples of molecules include glucose, amino acids, sodium ions, chloride ions. Those ions cannot cross the cell surface membrane. So they need the help of proteins. Which proteins? Are of two different types. So we have what we call the channel proteins. The channel proteins, for them, they have a channel. They will hear the word channel. So they have hydrophilic channels where the polar molecules can pass. Now, these proteins, most of the time, are specific. They are very specific. They allow only one type of molecule to cross. So a channel protein that allows glucose might not be the same channel protein to allow chloride ions. The channel proteins are really very specific in nature. So they create channels where those polar molecules can pass. Then some of those channel proteins can be gated. When someone mentions that the channel protein is gated, it simply means that part of it has a molecule or a structure, I could call it a structure that can cover the channel or that can open the channel. So sometimes the channel can be opened or it can be closed. So here, if it's closed, the structure will cover the channel so that no ions can pass through. If it is open, then the structure will leave the channel so that the ions can pass through. So sometimes the channels are gated. The way you can have a gate at home. Sometimes the gate can be closed so that no one enters, but when you need someone to enter, you can open it. So that is going to allow control of exchange of ions, in that not all the time ions will be entering into the cell, or not all the time ions will be leaving the cell. And usually those gated channels require energy to operate that gate, which closes and opens. It requires energy. So channel proteins allow facilitated diffusion to occur. This is a diagram showing us a channel protein for sodium ions. So this channel protein, for example, right here, it is open so that we have sodium ions entering into the cell. At another time, we can have it closed so that sodium ions do not enter into the cell. So that allows the cell to control exchange between itself and the environment. So those are channel proteins. You also have what we call carrier proteins. So carrier proteins usually do not have a fixed shape like the channel proteins. Channel proteins have a fixed shape. A fixed shape is just a channel. But carrier proteins keep on changing shape. They flip between two shapes. Like an example, if you look at this diagram, you can see this carrier protein is facing upwards. We have these polar molecules that come attached to the carrier protein, and then they are going to be sent to the inside of the cell. But for them to be carried to the inside of the cell, the carrier protein changes shape. Right now, you can see it's facing downwards. So if the carrier protein does not need energy to change shape, then we can say it is carrying out facilitated diffusion. So some carrier proteins just change their shape spontaneously. The moment the polar molecule to be allowed to enter into the cell surface membrane attaches to it, the molecule just changes shape spontaneously. It doesn't require energy. So if it doesn't require energy, we shall call that facilitated diffusion. However, there are some carrier proteins that need energy to change their shape. So if a carrier protein requires energy to change its shape, then that will not be facilitated diffusion. That will be active transport. So please note the difference between the two. This is still a carrier protein carrying out facilitated diffusion. You can see we have our polar molecule. It comes, attaches to the carrier protein, and then the carrier protein changes shape and pushes it into the cell. But what we are noting about this process is that the molecules are being carried from their region of high concentration through the cell surface membrane to the region of low concentration. So that is still facilitated diffusion. So usually the rate of diffusion through the channel and carrier protein can depend on certain factors. It will also depend on the concentration gradient. We already looked at that factor. So the steeper the concentration gradient, the faster the molecules will be able to cross the cell surface membrane by facilitated diffusion. But we can also look at how many channel proteins are in the cell surface membrane. The more the channel proteins, the faster the rate of diffusion. Or in that case, we can also look at opening, because we have seen some of the channel proteins have gates. So the more gates that are open, the faster the rate of diffusion. If the gates open are very low, then the lower the rate of diffusion. The lower the rate of diffusion. So that is it about diffusion. If this video has helped you understand diffusion better, give it a thumbs up. Don't forget to subscribe to my channel. I'm always going to be uploading notes for you, uploading videos for you, explaining the different topics. Thank you for your time. See you on the next one. Bye.